All right, everyone. Hi. The name's Nat, and welcome to Exceptional Individuals. Today we are going to be looking into the origins of autism. Where did it come from? Who are you? And we're going to be looking at it from a cellular level. Today is going to get quite maybe difficult in places, or definitely there'll be terms that you've never heard of. I'm going to try and keep it as top level as I possibly can, but if there are things that you want explained further, please do. Today is going to be under the assumption that all of you know what autism is. It's a neurodevelopmental disorder, and it can affect people with their social behavior, how they interact, sensitivity levels. It can affect repetitive behavior. Now, we know what autism looks like, but we don't really know what it is under the hood. And today we are going to the very beginning. With that, do know that this is an emerging area and it's a really complex subject. I've done my best to make it as digestible and easy for all of you to understand as possible. Today, we are predominantly looking at autism. It's good to know that a lot of the origins are quite similar wherever we're looking. Here's our lovely team. Just so you know, you got myself in the chat now, and I'm doing my master's in neuroscience and also neurodivergent myself. We also have April, who's doing the recording and editing today, who'll be able to help as well. Now, the last webinar we have that is uploaded, because we're still editing the other one, is the Blueprint of Autism. This is again looking at the foundation of autism. Do go onto our YouTube channel. You can just type in Exceptional Individuals and fingers crossed it will come up. But we do record all our webinars and you'll be able to see me chatting away, hopefully being all chirpy, teaching all the genetics and the brain stuff. And April has kindly done all the little chapters to make it really easy to find every section. Now, the complexities of autism, unraveling its origin. As I said, it is very complementary to understanding the treatment. Now, just so you know, there isn't like a silver bullet. When we say treatment, we don't talk about curing. We talk about how we can get the best out of someone and for them to live a fulfilling life. That being said, autism is still different to those who do not have autism. And that needs exploring. What is going on? Is it a natural part of being human? Is it when something doesn't go right? There's so many unanswered questions, and we are going to attempt to answer some of them today. Some of the overviews are things that we're going to be diving in deep on. We're going to be looking at theories on how the brain works. Ultimately, they're just theories. We do not know. There's some things we know 100%. And there's other things we're still learning. Research, mice, studies, and ASD. We are going to be looking at how mice are helping us learn. We are also going to be looking at a gene called SHANK3. This is going to be crucial for today. Out of curiosity, let me know in the chats, have any of you ever heard of SHANK3? No, I wouldn't blame you. I found it funny. I always maybe not funny, but I always remind myself like it's when you're getting stabbed 3 times, like shank. That's how I remember it. But it's it's a really crucial gene when it comes to autism. So it's worth one to look out for. We're going to look at how we're actually finding this information out. Do we have to slice and dice the brain up? Can we just scan it? Are there other ways of finding out what autism actually is? If you were to cut my brain up, would it clearly say I'm autistic, or would it still be a little bit ambiguous? Looking at the changes of people with autism, is there difference in terms of how their brain uses neurotransmitters, so chemicals, and also the different behavioral changes that you might expect to see in someone with autism, and also the mouse with autism? Can mice even have autism? That's something we're going to be looking at today. So my first question to ask to all of you is, why is it tough to pinpoint what autism is? Why do we still not know? How come we have fancy AI? How come we be able to go to the moon? We're able to cure types of cancers. We're able to do many things. We have an entire world of knowledge on our tiny little phones, but yet we still don't really know what autism is. It's only recently been discovered. Well, yeah, kinda. It's only in the last 100 years it was even conceived of, and since then it has changed dramatically over time in terms of our understanding. We have reclassified it, we have jigged it around, we have added things. At the moment, the way we diagnose autism is through the DSM, it's called a manual of mental health. And that we only diagnose it from appearances, through viewing behaviors, but we don't look from a brain perspective. It's hard to diagnose in the first place. It's very broad. Absolutely. It's spectrum. A spectrum means it doesn't fit nicely into category. There are some broad things that glue us all together, but really every single person is an individual. When we say everyone's an individual, what are we saying? Well, are we saying that everyone has their own personality? Well, yeah, but what is a personality? Every single thing, what's the difference between ADHD and autism? What's the difference of having a preference for spicy food to not? What's the difference between having ginger hair and blonde? It all comes down to a genetic level. And there are some things that when they do not go to plan, an outcome comes as a result of it. But why is it that all these outcomes are different? If you attended the last session, you'll realize that autism is a complex condition. It means there isn't one single reason for autism. It is a combination between a multiple of genetics, and we're not talking like, a secret autism gene. No, we're talking really common genes and lots of little different things in it. And it is the combination of them which presents itself as autism. There are many reasons why it could be different. If the brain was simple enough to understand, we wouldn't have the intelligence to understand it. That's one to make you think, but true. Yes, a misdiagnosis and lack of knowledge over the years. That is true. Originally they thought autism was just schizophrenia. They thought it was madness. They thought it was only for males. No wonder why. Maybe we've been barking up the wrong tree for a decade or two. A lack of genuine care and insight from government level. That is a completely different way of another dimension to look at this. But yeah, we haven't bothered putting the funding into it. Why? Maybe there's not enough people caring. Maybe there's just not enough insight or money. It is true though, money does get results when it comes to research size. It's easier to get into space than to figure out why someone feels spaced out. That's interesting too. So why do we not know the origins of autism? Well, money is always an issue, but also this really is incredibly complex. Now, please do not think I think autism and cancer are anything alike. They're not. But why I want to mention it is when we talk about, say, why haven't we cured cancer, is because cancer isn't one thing. It is many, 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 many different things. And autism is quite similar. It is a really huge spectrum. But the difference is we have cured essentially some cancers and illnesses. But with autism, we've not been able to. Now, the debate whether or not it is something that should be cured or not be cured is not really for today. Though it's worth noting that exceptional individual's perspective is that it is a difference which is valuable to society. However, I'm not going to let that influence today as best as possible. I'm going to use the quote, if the brain was simple enough. Yeah, I like that a lot as well. So this will give you an idea of how complex our brains are. They are ridiculous. So we've got one human being, and the genes in the human genome, 20,000, that's a lot. Cells in a human body, 75 to 100 trillion. And chromosomes in a human cell, 46. One of these things could be the reason for autism. One in a trillion. We have more cells in our body than known stars in our solar system. Our bodies have so much going on. We do not have the resources at the moment to go for every single one. All right, great. We know the human body is complicated. Do not expect a hard answer today. Out of all these million types of genes, we are going to be looking at one today. One out of a few trillion. Are we going to hit the nail on the head? Probably not, but it's definitely one to keep an eye out for. So what are some terms that we should probably know about? Well, neurons. Neurons transmit info through signals in the brain and the nervous system. Essentially all the cables in our brain, when one cable talks to the other cable, every time you blink, you think, you thought, every single thing you do in your body, it happens in the brain through neurons. You've got your neuron, and you've got your dendrites at one end, you've got your synapse at the other end, and essentially it's that communication. We've known for a long time that these communication cables are typically the reason why we have certain conditions, particularly neurodevelopmental. Synapse, as I said, they are on the end of the neuron, and these are the junctions before they transmit to the next one. Now, where the neuron will typically be like an electrical signal, the synapse at the end is a chemical signal. So it goes like electrochemical. Then we've got our neural network. This is processes in recognizing patterns and facilitating various cognitive functions. So if you have enough neurons together, they build up a bit of a network. And let's say you want to blink, move your arm, you'll have certain like highways. Think of them like express routes, and they'll clump together. Just like building muscle, I don't have any to show, but let's say I did, you could build up your neural pathways. These networks can get stronger. This is why you should also exercise the brain. So what exactly do we know those terms? I don't have time to go into them into detail today, but let me know if you want a bit more explanation. We need to understand, what do we actually know? We know that genetics, essentially the building blocks of who we are, and our synapses, they are linked to autism. And autism is essentially our behavior. So on here, let's say we've got our cells. In our body, we have lots and lots of cells. And in those cells, we have chromosomes. And within those chromosomes, we have DNA. And within the DNA, we've got the genes. As you can see, when we talk about DNA, we talk about it all the time. But it is really, 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 really tiny. And there is ridiculous amounts of it within our body. It really is a needle in a haystack in terms of what we're looking for. But in order to actually find that needle in a haystack, we're probably going to need some nice gadgets and gizmos. What do you think is the best possible way in order to do a little bit of digging and to understand the brain? Do we want a post-mortem brain tissue? That's essentially a dead brain. Like, yup, I'll have that brain. Slicey dicey, autism. Or is it fMRI, where we go on a machine and essentially we get a nice little image? Or is it a Polaroid where you go, oh yeah, clearly it's not a Polaroid, even though that would be cool. But let's see what you'll put. Oh, interesting. It is both brain tissue and fMRI. Truth be told, having an actual brain is actually really good and arguably the best. Why? Because you can get up close and dirty with it. You can twiddle with it. You can poke it. You can cut it up. There are no barriers. When we are going in a machine, essentially it's going to have to use like X-rays, different methods in order to see under the surface. And it means that the resolution isn't going to be that great. But what is the biggest issue with getting ourselves some juicy brains? Nobody wants to give them. We're like, oh, sorry, need that brain. And when people die, they don't really stay, juicy for long. Getting to do research on actual real autistic brains, it isn't easy. And arguably, there's some ethical things to consider. More often than not, we use fMRI. And this is non-invasive. Any of you can have it done. I know some of you previously have said you had it, and it can be a little bit scary, really claustrophobic. And let's face it, it's not the most autistic friendly environment. But these are the best things we got in order to understand what our brain actually looks like. Now, the benefit of using fMRI is obviously the brain is still alive. You're able to see what activations is and see it in a real world setting. As autism is likely about how information is sent from one point to the other, it's a very active process. Maybe a dead brain isn't really going to be able to show us that. They'll be able to give us some clues, but probably not all the clues. When we are looking at the brain, we do notice that the cerebral cortex is a bit chunkier in those with autism. But where is this cerebral cortex? Put a little pin on where you think this cortex is. Wow, okay, we are all over the shop. Anyone else? Where is this? Here's a bit of a trick question, by the way. If you're like, oh, I don't know, this image probably isn't the best image in order to show you. This cortex, this name might seem a bit unfamiliar to you. When we talk about like white and grey matter, it's like that. So essentially, it is the entire brain. It is the outer layer. It's like the ridges. So essentially, it is all over. The cerebral cortex is the outside of the brain, not particularly any particular area. Now, in those with autism, they have thicker areas. And this is what we can learn from when we're like looking at dead brains. I know dead brain isn't the correct term. I just find it funny. So there is an actual physical difference in those with autism. Yeah, it is a trick question, sorry. Now, in our brain, let's have a look. So this is if we're cutting it up. We could get donated brains, but, they're hard to come by. Not everyone wants to donate their brain for like some spotty uni student to poke at it for a few hours and then throw it in the bin. I don't know what they do. So we've got the top layer as the skin layer. Then we've got the bone. That's useful. Then, we've got also like the fiber and like the hairs and stuff. And then we got the cerebral cortex. You see the rigid, how it dips in and out. When you look at a picture of the brain and it has all these like ins and outs, that is essentially what we're looking at. And it is this little layer, which is thicker in those with autism. So I think I might ask you later on anyway, but do people with autism have bigger brains? I've asked you before, and they do. Autistic people have bigger brains. I should probably note here that bigger brains don't necessarily equal more intelligent, but it's fun to say. Einstein, for example, had a smaller than average brain, but it's interesting. It doesn't mean intelligence. We just have bigger brains. And I'm not talking like, noticeably bigger. We're not like bubbles, but tiny bits bigger. And some ways we know this is through doing research. So a person called Stoner, which let's hope he wasn't stoned at the time, did some really interesting research. And on this outline, you can see the different layers, and you'll see there's parts of the brain where you can see how thick they are. So there are lots of scans, and we've got the upper cortical layer disruption. If you notice, while there are thicker areas, there are some parts which just haven't formed correctly or seem weaker. There is something which is different to a neurotypical brain. At the moment, this isn't telling us what autism is, but what it is saying is that there is definitely something there. It isn't purely psychological, it is also physical. And the key difference to what we are seeing in these brains that are larger is a higher density of neurons. Remember, neurons are essentially information cables. They get information from one part to the other part. Now, that's like a good thing. Surely if I have more cables in my head, I'm going to be smarter and I'm going to be quicker on my feet. Imagine like if you got one internet cable for your whole entire town, it's going to be really slow. You have lots of internet cables, super fast broadband. But why is it that if people with autism have more neurons in their brains, that they struggle with processing rather than find it easier? Yes, they are harder to filter. Good point. That is something we're going to be looking at. I've already answered this one for you. I knew I was going to ruin a question. Do autistic children have bigger brains? Hopefully you will get this one right. They absolutely do have bigger brains. But do remember, it's not necessarily a positive or a negative. Autistic kids have more neurons in the prefrontal cortex. Now, just for fun, show me where this prefrontal cortex is. What part of the brain? And this isn't a trick question, by the way. This is, a real one. Oh, yes. The dots are coming in. It is at the front part of the head. In this part of the brain, which is very useful, we know that there are more connections here. Interesting. Yeah, sorry, Gregory. I am literally abusing the brain today. Stabbing, slicing, pinning it. Oh, science. So, okay, we've got more neurons in this area. Now, the really interesting thing is understanding why does more neurons not equal higher ability? And as some of you have already alluded to, too much info, it's harder to filter. Essentially, think of the brain like a plant. Now, plants will grow lots of shoots, but when some shoots, when some shoots like start to die or they're like not in the best condition, you'll pluck them, you'll get rid of them, and you'll chuck them away in order to let the new shoots grow up. If you keep too many of them, essentially they take all the resources away. It doesn't equal better. And that's what's happening in the autistic brain. It's very natural for something which we call synaptic pruning. Pruning, is when you're doing a bit of gardening. Look, the brain, some beautiful little neurons everywhere. The brain's like, you know what? Let's have a bit of a spring clean out. Let's get rid of and they'll chuck them away. But those with autism, well, for some reason, we're hoarders. We don't like chucking things away. We'll keep, it's true, and we'll keep all those neurons just in case of a rainy day. But that baggage is essentially not really helpful. Cutting unneeded connections during growth is a natural and healthy part of a developing brain. Now, knowing what I've told you, look at these little images of the dendritic spines. Which one is an autistic brain? Now, remember what I said. The dendrites are the front part of a neuron. Then you've got the axon, and then at the end, you've got the synapse. So the dendrite is the part that connects it to the synapse. Think of them like Lego. Now, which one is the autistic brain? Now, amazingly, you've all got it correct. How? Like, I'm curious to know. But it's actually, maybe it's quite obvious because I talked about how autistic brains are thicker and also they do less pruning. If we look at these images, and I appreciate it's like tiny, but you see how there's more little like knobbly bits on the end. These are called dendritic spines. At the end of a dendrite, you will have like little hair, think like hairs almost. They're little things that plop out. The more surface area, the more connections they can have. Now, people with autism have more of these little spines. So dendritic spines are small protrusions found on the branches of dendrites, which are specialized structures of neurons. It sounds very much like gardening. But yeah, if a tree had too many little branches, the nutrients aren't going to the parts which are needed most. Now, this is how it should be done. In an ideal world, this bit of pruning would work. You've got a tree and, okay, the branches have grown. Then we do a little bit of pruning, we trim them up. We make sure that only the finest brain cells are flourishing. We remove any brain cells which are just not pulling their weight, aren't that weak, are taking away precious resources. And then the connections between our brains, while there are less of them, they are more efficient. So again, like muscles, they've gone more stronger and they are more adaptive. It's much better to have less but stronger than multiple and weaker. Whereas if you've got unusual pruning, let's say in autism, it doesn't work like how we want it to work. Again, we got our branches, the pruning isn't happening, the tree's getting a mess, no one's cut it for ages, the gardener's on holiday, and then we see there's lots of improper synapses. These connections, they're weak, they're dodgy, it's faulty white wiring, and that equals a higher risk of ASD. Now, it's not like we can just hire a gardener and say, get your ass here now, need some pruning done. That's beyond our control. But it does help you understand why this is neurodevelopmental. It happens either in the womb or very early on. When your brain is busy sprouting all the new shoots, which are going to be with you pretty much for life, this is a crucial part for the brain to say, all right, get rid of that, have a bit more of that, let's make that stronger. Oh, let's keep an eye on that one. But what else have we actually learned from these tissues? We learned about these dendritic spines. But are there more things that are worth keeping a cheeky eye on? Let's spin the magical wheel. Oh, we've also learned that animal models are really useful for studying autism. Why? Well, we talked about how brains are actually really useful for studying the brain, but people don't like giving their brains up. Animals, however, they love giving their brains away. Well, Emma wants to give her brain away. Okay, cool. Emma will give her brain away. We'll keep note of that. We'll wait for we'll wait patiently. I hope you stay alive for long. Animal brains are really useful, but not all animals. Certain animals are more closely related to humans than other animals, but also some animals are not that rare and really easy to get hold of, which are better. Oh, what other things have we learned? Come on, spinny wheel. Lost neurons in cortical layers. Oh, we also learned that sometimes they can just disappear. Let's have another look. I have to use the spinny wheel because the text is small. ASD varies, affecting research. This is one of the bottlenecks, is while we will find some consistent patterns across the spectrum, honestly, the results are not conclusive because some people have something, another will have another thing. There isn't enough concrete evidence which is consistent amongst enough people in order to make some really strong conclusions. Lost neurons, and there was probably another thing, but the spinny wheel may never land on it. So let's look at some animal studies. Animals are going to be absolutely crucial for ethical reasons, we cannot be slicing and dicing people up every 5 seconds. They used to do that in like medieval times, but even then it was frowned upon. But people would just be walking along the streets and a body snatcher will come and take them away. They'd kill them up and they'd sell them for a couple of bucks and we're done. That is honestly happened in London. Grave robbers would literally dig up bodies when they had just been buried to take the brains out to give to researchers. A great time for science, a bad time for being a person or a corpse. So animals, it is them. Some say it's the reason why unicorns don't exist anymore. Their brains were too close to autistics. I don't know if that's true, probably not. What do you reckon is the best animal model for ASD, Autism Spectrum Disorder? What animal do you think is the best for support for using as a base model for autism? Okay, nice. Okay, easy. This is interesting. Most for mice, one for monkey. Now, arguably, monkey would be better because monkeys are closer to humans. The biggest challenge with monkeys is we didn't look after them enough. We've killed them all off. There's not enough of them. And they're really, really smart. And it's just, monkeys aren't going to do it. But yes, mice. Now, you could argue that there isn't a best animal. It's the best animal for a particular study. We use zebrafish quite a lot to study conditions. Why are we using a fish? Well, because they're transparent and we can actually see their brains working while they're alive, which is really cool. But mice are used for a few reasons. One, they are plentiful. You're not going to run out of mice anytime soon. Two, they breed really, really fast. Because what you want to see is how mice replicate during generations and how it affects them through like 5th, 7th, 9th generation. We also use mice because they're quite good and easy at replicating to keep a level of consistency. But you can also use rats, you can use rabbits, but mice tend to be the animal of choice. Bear in mind, I'm a vegetarian, I don't condone animal violence, but the animals have genuinely helped us a lot. The next time you're like, oh, a mouse or a rat, know that we owe them a lot. Moving on from animal models, let's look at some genes. And I'm not talking about those denims that you're wearing. I'm talking about the building blocks of what makes you you. Synapses, genes, and ASD. We are getting to the origins of autism. Oh, Chris, that's definitely not true. The unicorns have all learned to camouflage better. That's nice. But again, just like we are very much like unicorns. We talked about genes, and I talked about SHANK3. SHANK3 isn't just some person going around shanking people. SHANK3 is a particular type of gene or protein in the human body. Now, with most people with autism, we've noticed that this tends to be a gene which malfunctioning, isn't working how we're expected. There's stuff definitely something a little bit iffy going on with SHANK3. And this has been an area of keen interest. So you can see the two differences there. But we what exactly is SHANK3? It is a protein, but essentially a gene that is crucial for brain cell communication. If the communication isn't working, have you tried restarting your SHANK3 gene? That could be a reason. But let's have a little look. We've got neurons, and in your brain, you've got your neurons, and in your neurons, you've got genes, and those genes have proteins, and those proteins, some of them are SHANK3. Here's a little image of a pre-synapse and a post-synapse. And these are the bits, where you send your chemicals across to get the messages. And in here, you've got your SHANK3 in the post-synapse. And this is an area which is problematic to say the least. We don't have that much information. It is definitely an area which a lot of researchers are really interested about. Could this be the OG? Maybe, but probably not the entire story. Here's an image which might help you understand it a bit better. In our bodies, we have our DNA and our genes, and the SHANK3 is kind of like scaffolding. It holds it all up. But when the scaffolding isn't working right well, the genes don't have the same structure, and potentially autism comes as a result of this dodgy scaffolding. So SHANK3 equals scaffolding for glutamate receptors. Now, we know glutamate is a neurotransmitter. It is one of the chemicals which allows our bodies to function, makes decisions. We've also spoke about glutamate potentially being the OG reason for in comparison to dopamine. But glutamate looks like it has quite a stronger relationship when it comes to autism. So detection, when we have found SHANK3 deletion, we have seen more likely causes of autism. Deletion is when something should be there, but isn't there. SHANK3 is like scaffolding. When we remove the scaffolding, the building falls down and you have autism. That is one way of looking at it. Please bear in mind, this is a really simplistic way of describing it. Hopefully it's enough to get that imagery in your head about why we have autism. We have autism because someone didn't do their job and didn't put the scaffolding up. Personally, I love being autistic, but it's still interesting to know. So SHANK3, is it actually connected to autism? Well, in our genes, we've got the 22nd one is the potentially problematic one. We know that it is a key cause of autism when deleted. It doesn't mean it's purely that. So there's more to autism than purely the SHANK3 being Control-Alt-deleted, but it is definitely something which we are keen to learn more about. We know that it is a big impact gene. There are many, many genes. Some are more important than others. SHANK3 tends to be a big player in the game. And we also notice that it is associated with Phelan-McDermid syndrome. This is a syndrome which is very closely linked to autism. So there's more research connecting this than there is to autism, but this is also very closely linked to autism. Okay, to summarize, because I appreciate this is quite a lot to take in if you've never done any neuroscience ever before. To summarize SHANK3, and Dory, it's not like SHANK3 is like the new and improved SHANK2. They are just different. SHANK3 proteins create and keep connections. So rather than seeing it like a rigid bit of scaffolding that gets like rusted over time, it is scaffolding that is always being replaced, always making sure that is up to code and making sure everything is working swimmingly. It aids in cell communication. A good shank equals good communication. Changes can cause poor communication, and this can lead to autism. You've got to admit, this is looking pretty promising for the origin of autism. Pretty promising, more to know. As you can see, we've got our pre-synapses here, our neurotransmitters are coming out, our receptors are taking them, and then what happens when our body has taken in these neurotransmitters? Well, before they get to the next postsynapse and dendrites, they go through the shank. And if the shank is there, good. If it's not, maybe there's some other things like issues that might come up. Moving on nicely to pinky in the brain. To study SHANK3 and autism, a knockout mouse was made. Now, a knockout mouse isn't like the Rocky Balboa of the mouse world. Essentially, it is a man-made mouse. It is a mouse where we went into the circuitry and we started flipping all the switches, turning some genes on and some genes off. It isn't absolutely a real mouse, but it has been perfectly altered in order to mimic some things working and some things not. Now, remember when we talked about how many potential genes there are in a human body? Well, animals also have a lot of genes. So think of it like you've got loads of light switches. One light is turned on, but you don't know which one. You turn one light on, okay, not that one. You turn that light on, and you go through all the different lights. But way more complicated. Now, while we have genetically modified a mouse in order to have SHANK3 active or not active in order to see if we're seeing the same attributes of autism, can a mouse have autism? Okay, we've got yes and we've got no. More people think yes and no. Now, the truth is no. A mouse cannot have autism. Why? Because autism is a human condition. That being said, it can have autism-like conditions. It can look very similar to autism. Not a one-for-one comparison, but there is definitely something there which is still close enough, which still makes it useful and interesting for us to observe. Let's look a little bit more at this knockout mouse. And I always like the knock-out mouse. I just like a mouse getting a bit of a punch up. But all it means is a mouse that has been genetically modified. To be fair, you can get knockout rats, knockout rabbits. You can get knockouts of most things. It's something which, and what they'll do, they'll create a knockoff mouse and they'll keep breeding it and seeing how, once you have altered one particular gene, what happens to this mouse's behavior in future generations. And this is very useful because we know that autism runs in families, is hereditary. And if such, is it also hereditary in mice? Well, yeah. Well, at least the characteristics of autism is. What are some of the things we're looking at? There was a study done in 2011, and this was to observe mouse behavior of knockout mice, which have had the SHANK3 turned off or on. They found that mice acted like they had autism, had big striatums. So while not directly autism, a lot of the characteristics were very similar to autism, and all of those mice had big striatums. This is interesting, right? Because we know that autistic children have bigger brains. And mice who have similar characteristics of autism, well, they've also got a few more neurons kicking around. The conclusion was knockout mice does aid in understanding autism. I know, a bit underwhelming. It didn't actually tell us any answers, but it did tell us that there is enough similarities that this is an interesting place to investigate and dig a little deeper. The next study, let's look at mouse's brain connections. We found out in this study that smaller synapses, smaller connections were in the mutant rats, or the mutant, knockout, the same. Reduced synaptic function, the communication wasn't as good. Knockout mice had complex dendrite branches. Well, isn't this interesting? Because when we were looking at autistic children's dendrites, we saw that their dendrites were a little bit more bobbly. There was more stuff going on. They had less pruning. And the mice have the exact same look. So this image is a bit confusing, but essentially there's more going on when we're looking at the mice that have SHANK3 and the ones that do not have SHANK3. Mice genuinely are a useful tool. That being said, though, mice aren't perfect. So what do you think is a big issue with knockout mice over just using humans? We talked about why humans aren't always great. Why are they still better than using mice overall? Mice aren't humans. It is very true. Genetic modification, social communication. Yeah, there is a particular answer I'm looking for, and maybe I'm not expecting you to get it, but these answers are still correct. And it is not a one-for-one comparison. They mimic certain instances of autism, those ways that we say they look autistic, more of an observation. For instance, when a mouse is being less social or choosing not to interact with its peers or has less interest in certain areas, that is what we mean when we say autistic characteristics. We don't know that for certain, but the answer that I'm looking for is knockout mice have complete removal of SHANK3. In humans with ASD, it is heterozygous disorders, which is the loss of only one copy of the gene. Other copies remain present. Okay, let's break that down because I can see that already. Heterozygous disorders is when you have one normal gene and one mutated gene. So as you can see here, a gene, you see you got one here. Let's say, for instance, this is the SHANK3. On the other side, there would also be a SHANK3. Now, we would get rid of one, but in mice, you get rid of all of them. So it really isn't a direct comparison, even on the micro scale. Mickey Mouse has rabies. The heterozygous mouse is created. And this is a mouse which mimics more closely to how humans experience autism. We've had to really manipulate these mice to look way more like humans than they would previously or naturally. So one changed gene in a mouse equals ASD-like behavior. You notice how I'll just go quiet when I'm saying the word. You didn't hear me properly. Equals fewer hippocampal synapses. SHANK3 lost equals synaptic function problems. Even these mice are mutants at this stage. They are not normal mice, but they are starting to look more like the individuals with autism than any previous models. It's all adding up at the moment. Now, the key thing to notice is we today have looked at one particular model. There are over 70 mice models that exist when it comes to monitoring autism. And you can go online and you can buy yourself a knockout mouse that has SHANK3 turned off. You could also go and buy a mouse that has SHANK2 turned off. So don't go and buy these as pets. They're probably not great pets. I've got myself a little autistic mouse. There are many, many different types of mice, and there are lots and lots of studies. We've only looked at one. Today, we haven't found the origins of autism, but we have found we are on the right tracks, and we are getting closer and closer. What is it? Is there a consensus? Is everyone in agreement that we appreciate that SHANK3 is a likely contender? No, there is still a lot of debate. Let's go through this. First of all, E/I balance is essentially the excitation and inhibition balance, and TD equals typical development. Changes are common in mouse models. When we have changed the mouse models, we notice that the side areas work worse. Changes can go both ways in different synapses. Both ways can change in terms of how information is passed to the inhibition balance theories and more. Currently, our findings are matching up with preconceived theories, and the balance input is linked to ASD. It's all linking up at the moment. Our research, our studies, our theories and hypotheses are linking up. And as you can see here, depending on how the excitatory and inhibition balances go in male and female, if it's typical development, normal social behavior. If it's atypical development, then you might get diagnosed with ASD, and that's when you'd have different social behavior. So it is at this area where it is either you have autism, you don't have autism. And you know how we talk about autism as a spectrum? It's not like yes, no. Think of it like this image, like a scale. And some people will be quite balanced, other people will be a little bit off balance, and some people will be really off balance. And depending on this excitatory and inhibition balance will really dictate maybe the level of needs of autism that an individual may have. Still a theory, but still interesting nevertheless. Mouse models, usefulness for human ASD is debated. We talked about why mouse models are great. We've also talked, fireworks. We've also talked about why mice, let's try another one. We also talked about why mice models are bad. Yay, thunderstorm. All right. Mouse model usefulness for human ASD is debated. Growing human brains in labs research is ideal, but is it ethical? We've learned that mice models are really good, but they're not perfect. And the debatedness isn't just about animal rights. It's also the fact that mice are not human. And if they were like humans, we probably can't operate on them. The next best thing, or the better thing, would be to use human brains. But we can't be stealing brains from people. Maybe we could grow a brain. Is this ethical? Yes or no? Oh, a lot of you have said no. I don't know where I feel on this, because surely if you've grown a brain, doesn't it not then become a person? Or does it not? Like, I would need to know more information. Like, is this brain conscious? You know, it might not have a body, but is there more than meets the eye? Are we then playing God? The next question is, even if it was ethical and people were okay with it, do you think it's actually possible in order to grow a brain? Yes or no? Okay, we got yeah. Now, heads up. Oh, thanks, April. It is not possible at the moment, though we have tried. We have things called stem cells, which essentially we can, in petri dishes, we can have cells that can be anything that we want. We're not there yet. We're not even nearly there yet. But in theory, it would be possible to create something which looks very similar to a brain, and you could still run different tests on. It wouldn't be like a full squidgy bit of meat, but maybe more like a petri dish where you can see how the synapses, the connections are. Overall, what did we learn? And now I need you to say if you disagree or if you agree, if these are the things that we've actually learned today. Because remember, the debate is ongoing. Did we learn that synaptics problems are in ASD? Well, I think we did. We noticed that people with ASD do have problems with their connections in their brains. We also learned that inhibition balances matter as in ASD. Yeah, I would say we know that. We also learned that brain cells can be impacted in terms of like the pruning. There are things that we definitely know about autism. We know more than we have ever known, but we are still quite a bit off from fully understanding all the tiny little intricacies about what exactly autism is. Just to round up with, the key takeaways today is when we're looking at genetics, ASD is linked to genes. It is a genetic condition, but it is a complex condition. It isn't one tiny gene, which is the issue. There are potentially lots of little genes. And scientists are looking at individual genes and then trying to build a bigger picture to have a look at the whole individual. We are not at a place where we can scan someone and say, that's the gene. It's that cheeky SHANK3, which is the one to look out for, or it is that one or that one or that one. We also know that brain abnormalities can have unique changes in ASD brains. Some people might have different synaptic, different dendritic spikes, and other people might have other different ones. Slight tiny changes. Just, when we look at these little spines, they can be like that, they can be chunky, they can be thin, they can be wiggly. There's many tiny changes which can happen in a human body, which can equal to different ways of demonstrating behavior in real life. Hence why autism will always be a very unique individual condition. We know that SHANK3 is important. That pretty certain SHANK3 is important. We just don't know how important, and we don't know how much of the story is actually telling. We know that the E/I balance key theory in AHD brain signaling definitely is something which is looking promising. And in the lab, we are known that insights into ASD cellular basics are improving dramatically. There are lots of things that are going on. If you thought, what today was useful, I actually would love to learn more. Check out some of these references. This will be on the recording, but there are lots of things and papers that are going into this and more information. I'll be honest with you, they're not the easiest to read, but and if you ever try reading any of these reports, do know that you are not meant to understand them. The people spent their entire life learning this information. The way I read them is I read little bits and I don't understand something, then I'll research into that, and it's lots of little rabbit holes. So, for instance, if I want to just understand one paper, I honestly have to read like 10 or 20 of them in order to understand one segment. You do not read them like you read traditional books. Do not feel bad if you honestly have no idea. A book that I can actually recommend, which is very easy to read and easy to understand, is The Autistic Brain by Temple Grandin. Now, Temple Grandin is an amazing individual who has been a massive advocate. And this book goes a bit more deeper into the autistic spectrum, while also being very accessible for the average person without a scientific background. Now, the next webinar is not next week. I have cancelled next week, unfortunately, because I'm at a wedding and I'm really bad at knowing things in advance. I'm the best man. I should have known I was at a wedding. I know. I didn't know. I'm really bad at dates. I am not here next week, and I have rescheduled that webinar, but the week after that, in 2 weeks' time, we have Autism Biomarkers. And if you don't know what a biomarker is, then you should turn up. Essentially, when we are looking at where autism is in the body, like what it is, there are certain areas which are really, really interesting to look at. Let's have a look. We've got understanding autism's new ways to diagnose and treat. At the moment, we look at autism in terms of how it demonstrates itself. Could it be possible that in the future we could use scans and equipment and AI in order to diagnose autism? Well, we've got little markers in our brain which basically computers potentially could diagnose autism in the future. Here is our YouTube channel if any of you are interested in signing up, learning about more. For those who thought today was interesting, or I wish I attended last week, which was meant to be this week, click subscribe, go to our YouTube channel, and you'll see like our videos. April uploads it pretty much weekly, and you can see we've got lots of interesting videos that you can check out. And here is our contact information if you have any further questions or want to get in contact. And we are done for today. Thank you so much, everyone. All right, everyone, I hope you found that useful, and have a great rest of your Thursday. Thanks, Vicky. Thanks, Bobby, Paula, Gregory, Zia, Anna, Ali. Thank you all for coming.